Greetings, everyone. I'm extremely delighted to have this opportunity to discuss some high-yield concepts today as they relate to our conference theme of rare and genetic kidney diseases. My name is Ajay Srivastava. I am a nephrologist and senior medical director at Medpace. So today I very simply want to talk about three major concepts, all within the next several minutes or so, which are to just basically briefly talk about common aspects of rare kidney diseases, review key considerations in clinical trial design and drug development in this setting, and help to understand the patient perspective in optimizing trial success. So, as already noted during this conference, the definition of rare diseases varies across the world. In Europe, for example, it's defined as a prevalence of less than 1 in 2,000 individuals, whereas in the U.S., its designation applies to less than 200,000 people that are affected. Therefore, although it's deemed rare, the effect is still large, especially when you add up all those that suffer from these various diseases, as Vijay had talked about earlier. And certainly, it's always 100% in those that suffer from them. Now, for inherited kidney disorders, the diagnosis and management are complex, especially as they can result in multi-system complications. And patients will spend years visiting many different healthcare providers before ultimately receiving an accurate diagnosis of a serious disease process that's often chronic, progressive, and with high potential for fatality early in life. And although advances in renal replacement therapies have also included greater access, making way for prolonged patient survival, patients often continue with a poor quality of life. In particular, children born with severe congenital nephropathies have a high likelihood of altered cognitive, physical, and psychosocial development and need for complex care. And while therapeutic advances in rare kidney diseases have historically had notable challenges, including populations with highly heterogeneous and variable clinical phenotypes, as Vijay also mentioned earlier, limited knowledge of the natural history of disease, which impact outcome and endpoint measures, lack of biomarkers to monitor disease progression, oftentimes with data diagnostic classifications that don't necessarily reflect the underlying pathophysiologic mechanisms, they do represent a notable unmet medical need, especially in the pediatric population, with a requirement to continually advance strategies to overcome these unique challenges, as we're going to discuss. Okay, so as we know, the development of treatments for rare disease and orphan indications represent special and rewarding opportunities to make truly meaningful and innovative contributions to medicine and those that suffer from these diseases. In the next several minutes, and in giving to the high-yield nature of this talk, I'm going to run through these 8 important points on the left with the associated key considerations that you're going to see in the upcoming boxes that'll appear on the right as we journey through strategies to optimize clinical development. Okay, so as with any program in general, especially with those that have complex paradigms, outlining the clinical development program early is fundamental. And this includes having a solid scientific foundation, which really requires good understanding of the mechanism of action of the IP, characterization of the disease with a defined natural history, and therefore an emphasis on a data monitoring or natural history study started early. Additional population-based considerations are also key and require informed understanding of available therapies, what the potential for curative or mitigating aspects of the disease is, and navigating the course of disease in terms of its chronic or progressive and sometimes rapidly progressive nature, and whether there are exceptions toward expectations, toward remission and relapses, which add a further dimension to its inherent complexity and psychosocial impact. As alluded to earlier, we know fundamentally that those with rare diseases represent challenging populations with regard to recruitment and require important considerations. So pre-identification at appropriately selected sites is key. Usually, those patients with rare diseases are seen at academic institutions, specialized centers, and pediatric hospitals. and successfully working with them as well as pediatric research networks are fundamental, especially if they are already offering genetic testing for disease confirmation. Additional strategies towards selecting appropriate investigational sites include those that have a thorough understanding of the epidemiology and inherent patterns for the disease, those that have familiarity with the standard of care and are institutionally consistent, especially with regard to background care or medications. Having diagnostic and therapeutic resources available are also key. And so in this regard, having an understanding of the startup processes for these sites, including the development of study-specific recruitment plans and materials, and ensuring fundamental study site training is vital. So a site's database will usually be the main source of potential subject pre-identification, including information on genetic testing and biopsies. And with that, will allow for efficient enrollment optimization. Sometimes, depending on the disease process, partnering with an institution's pathology department is key, because many times, especially for biopsy-proven diseases, all paths lead to the pathologist, we like to say. So partnering with pathology departments too to identify potential cases can be important. So subsequently, after site selection, the development of recruitment plans allows the teams to evaluate any challenges to individual participation. Collaboration with patient advocacy groups, as Uptal had mentioned earlier as well, which have an abundance of information, including current clinical trials, can significantly enhance study awareness and understanding, and potentially increase both site and patient interest. Having understanding and ways to assess and address individualized concerns, such as the psychological considerations in the setting of those that suffer from the disease. And within that, family and social dynamics, and ensuring patients have adequate transport for visits, the impact of IP administration, which can further impact compliance, as well as ensuring proper site and subject IP storage are all key considerations. And in this regard, the provision of travel and lodging, as well as home health nursing, as well as properly describing at the level of the patient's understanding the key procedure sequencing, timing, and frequency of evaluations are all important from the patient perspective, some of which we're going to touch on a little bit later. And certainly anticipating the nuances of regulatory timelines between sites and institutions are key. Okay, so as we know, and as we've discussed earlier, studies in rare diseases quite often encompass pediatric patients, which represent a vulnerable population with dynamic changes given the expected physiologic changes that are expected, including growth and also changes in their understanding of the world. And so it's really imperative that the study team anticipates the complex operational, regulatory, and ethical considerations in this population, including those raised by the investigator and parents. Certainly, studies should be conducted in subjects who directly benefit from participation, and with the study, and with that, the study IP is typically evaluated in older, more fully mature cohorts, and then followed by evaluation in younger patient cohorts. Now, unless the risk is minimal or the study will yield information that contributes substantially to the benefit of other children with the disorder, as it may be difficult to ethically justify studies in young patients when there is no opportunity for the patient to directly benefit themselves, we find that, again, that we don't necessarily see them studied as often in younger patients until they're actually studied in older children. Now, these studies required designs that would optimize meaningful outcome data while also minimizing patient discomfort and risk. And so there must be notable justification for anything that increases safety risk, including sedation, radiation exposure, and or invasive procedures. And in general, the minimization of procedures also needs to be attempted in general. Blood volumes for lab sampling have to be anticipated, documented, and monitored to ensure compliance with the WHO guidelines. Fully informed consent has to be obtained from the legal guardian and patients. Should assent to participation is determined by ethics committees or local legal requirements attest to. And as we very well know, communication differences based on age and development will impact both the content of assent documents as well as the conduct of the informed consent process, which may benefit from the utilization of pictures, videos, and child-friendly demonstrations to help explain procedures to younger children. And the ability to use rescue therapies during the trial, as well as integration of an open-labeled extension, will help with justification of a placebo-controlled design with regard to regulatory approval, and also help with the patient help with patient recruitment and retention as well. Okay, overall, with regard to approval, orphan drugs are held at least to the same evidence-based standard as non-orphan drugs. And in that regard, two common reasons for clinical hold from the FDA perspective are subject exposure to an unreasonable and significant risk, as well as an insufficient amount of information in the IND that's able to actually assess that risk. So therefore, common safety considerations include sufficient characterization of the drug or the biologic, and having sufficient pre or non-clinical data. For example, animal toxicology is important. Therefore, the successful translation of regulatory and ethical standards into study conduct requires a comprehensive understanding of the unique concerns raised by ethics committees, investigators, parents, and the patients themselves, which we'll discuss. Timelines for ethics committee reviews may prolong timelines if the rigor of their review is unanticipated. For example, ethics committees may want to involve pediatric experts if a member is not a pediatric expert, or may request to involve investigators who are experienced in working with children, especially if the study is not run at a site specializing in pediatrics. Ethics committees will inspect patient documents such as ICFs and other patient items, and they may require to involve a specific pediatric committee in the process to lower the risk of lengthy negotiations, deficiencies, and/or queries, knowledge of previous experiences with each site, and discussing the EC review process and expectations with each investigator prior to submission is important. Okay, safety monitoring, one of the topics I really enjoy talking about, and as we know, safety monitoring is one of the most fundamentally critical aspects of clinical trials, especially with regard to vulnerable populations. As such, safety aspects in rare disease clinical studies has to take into account a number of parameters regarding this unique patient population, and that includes lab monitoring, and not just for critical alerts, but conditional ones as well that are nuanced for both the age group being studied as well as the underlying kidney disease, so as to capture potential issues early before they become critical. Likewise, taking into account the unique physiology of the patient population, including an understanding of the common comorbidities and the interaction of these conditions in the context of the IP's mechanism of action, as well as the different age groups that are often more susceptible to particular toxicities due to age-related changes in physiology is key. Also of notable importance are that lab and vital sign ranges may need to account for the age, gender, and height and weight-adjusted norms excuse me, as necessary. Also, ensuring adequacy of the site's study equipment for assessments, such as appropriately sized blood pressure cuffs and scales that are calibrated, require proper review as well. So all of these considerations allow for the optimization of anticipated potential AEs, which in turn ensure rapid detection of safety signals, and certainly would need to account for study-specific parameters. And those parameters should address the needs of all cohorts and be interpreted appropriately in the setting of normal physiology. Also, automated calculations we found of percentiles and z-scores for parameters such as weight, height, height, velocity, and BMI parameters using EDC programming is very helpful for ensuring accuracy of data and avoiding unnecessary queries. Also, the use of visual analytics programs can help with signal detection, especially when reviewing complex data, which are typical of lab-heavy studies, especially when they're nephrology related. Also, these visual analytics programs can help plot those previously mentioned height and weight parameters, both individually and for the study population to evaluate trends. And finally, with regard to the study and population-specific lab alert flagging that was noted before, can be used as additional layers of subject protection by providing study-specific safety guidelines to investigators with provisions for additional diagnostic evaluation, background medication, and diet review, IP interruption or withdrawal, and or the use of rescue medications. Okay, and so we've arrived at the very important concept of reviewing the patient perspective in terms of clinical trial design in the rare disease space. One of the things that I alluded to earlier was that of the burden of trial participation, especially as it relates to addressing individualized concerns, such as the psychological considerations in the setting of those that suffer from the disease, including family and social dynamics. Ensuring patients have adequate transport for visits that impact, as well as the impact of IP administration, which can further impact compliance. The provision of travel lodging, as well as home health nursing services. And very importantly, that the subject and guardian understand very clearly the study procedure sequencing, including the timing and frequency of evaluations, which are extremely important, especially those involving invasive procedures and the number of blood draws. In that regard, early involvement of patient representative groups and organizations can be key by helping to evaluate these parameters in the setting of clinical trial development. Interestingly, expectations for outcomes can be age dependent. So, for example, parents of young children may have higher expectations for finding a cure than those of older patients who may be looking at more an impact on symptomatology and can affect study participation. Similarly, cultural differences can also affect participation, especially with regard to how they may view clinical trials or pharma companies in general. And having the patient perspective on certain trial design aspects as well can be key. So, for example, eligibility criteria where depending on the subject age, you may find difficulty in recruitment if PK analyses with multiple blood draws is a requirement for all patients rather than achieving samples from a proportion of representative patients. The use of placebo in certain instances may hinder recruitment unless, of course, as noted previously, an open label component is available, or potentially designing an open label sequential design where subjects can act as their own control. And certainly also comparisons to registries are an option, of course, ensuring that the appropriate amount of data is available for propensity score matching is also going to be important. And so coming back to outcome measures, while ensuring appropriate clinical trial duration to achieve an appropriate outcome is key, you may find that trial subjects may want a longer duration, especially if it can affect their symptomatology, which again is an added advantage of an open label component. Again, these are just some of the many examples one could use, and we at Medpace would be happy to speak to you more on those as well. So overall, we get to our last piece of the presentation, for which the overarching theme points out the importance of collaboration, which fundamentally is a key component of clinical trials, especially in the rare and genetic kidney disease space. And certainly this collaboration, communication are key components that we very much value at Medpace. I'm going to credit Carrie Shield. She's one of our really amazing directors of clinical trial medicine at Medpace. And last year, we actually gave a webinar on CKD and the impact on clinical development, in which she outlined a lot of these items that are on the slide here. And so, starting from the top, ensuring therapeutic and scientific oversight with medical experts, as we have at Medpace from project start to finish, is key with regard to clinical development and regulatory assistance, and most certainly, again, with patient safety, which I can't overemphasize enough, which needs to be considered at the very start of the entire process. And most certainly having close operational and medical collaboration is key in order to develop proactive and contingent planning with streamlined communication to address issues early to ensure trial success for all of the items that we discussed today and more. And enough cannot be said about the continued development and application of innovative and flexible solutions, which includes digital and tech opportunities, as well as the visual analytics that I discussed earlier, which also help reduce patient and site burden while also optimizing safety. If anything, patient and site support are fundamentally key, and ensuring that you have a partner or that you partner with those that have this fully addressed is extremely essential. And lastly, and importantly, trial oversight that ensures quality data and endpoint protection and analysis really can't be emphasized enough. So finally, we at Medpace, we really thank you and the organizers the opportunity to collaborate and speak today about the rare genetic kidney disease space. And certainly is an area that we are very passionate about, and we very much look forward to partnering with you to help you achieve success. So thanks so much.